Then we're going to go to our next speaker, colleague of mine, Nikita Ivanisenko, also the computer models to create different vaccines against different mutations of COVID. He's going to talk about the current tools based on a machine learning can be used for design based on protein, as protein, one of the mutations of the COVID key materials and the vaccines aimed at. Nikita, you can start.
Коллеги, добрый день. Семен, спасибо за представление. В данном докладе я хочу рассказать про нашу работу.
I'd like to talk about our work, where we have in our Institute of Artificial Intelligence, which we are using to get a more efficient anti-gene strike structure, rather than we do it today in most research. Why is SARS-CoV-2 is interesting about? This is a real relevant problem of today. This is the virus, one of the most researched in terms of the crystallographic data from which segment of S protein responsible for antibodies. So this is the best system to start working out the technology which would allow us to use rational ways, computer approaches and AI-based solutions to create prototypes of vaccines of synthetic antigen, which would allow us to develop antibodies and should be efficient protecting against the virus. The virus itself, SARS-CoV-2, goes inside the cell thanks to S protein, which are on the surface of virion, and they can recognize the human receptors, and while linking to human receptors, it leads to the merger in brain and goes inside. Due to the S protein fight, many vaccines are based, which are efficient, are based on using S protein as an antigen. In a Sputnik situation, there is a particular vector which codes S protein. It gets into a cell where it generates. It is generated, then there is a generation of antibodies, and when antibodies fight real virus, they protect. They just take this as protein function. But not everybody knows that antibodies can merge with different sites in the molecular structure in S protein. If we look at the crystallographic data where the antibodies go on the surface of S protein and we put it in green light, we can see that there are particular sites of for antibodies recognition. These are epitopes, and we have a lot of them. In fact, when Delta variant appeared, there was kind of a panic that the number of mutations happened on a key epitopes for antibodies recognition. And it was a big question, what is going to happen to the vaccines if we have some mutation going on? In epitope's case, but the problem was not that serious. We have a lot of epitopes, and even if one epitope is less efficient to generate antibodies, there are other epitopes for antibody generation. There's also T-cell immunity, which helps a lot. So that's a problem of vaccine development of new generation. This is a long-term problem, actually. In short term, we can use the current vaccine, but in long term, we believe that it's possible to try rational approaches, not when we only take the virus, protein virus and use an anti-gene, but we can create special structure using AI, using ML, and many algorithms which are becoming more and more developed. In this respect, there is a question whether we can do the following. When we take as protein and embed synthetic changes, or whether we can get a bigger number of antibodies and a bigger efficiency of recognition and resistance by increasing the number of epitopes which can be recognized. One of the approaches which can be applied to solve this particular task focuses on the following thing. This virus as protein is a very sophisticated molecular machine. So how as protein can evade immunity system, how it can recognize human receptors, how it can merge, it's a very true detective story. And it is important to understand how the virus works. But for the vaccine, this as protein analysis is not actually required. The only thing we need for this vaccine going into the body should generate antibodies which would fight against the real as protein or real virus. So we set a task in front of us. So let's freeze the virus in the way on the surface where there is the biggest number of epitopes. How can we do that? So we are lucky to be living in the 21st century. And there are more and more methods applicable for molecular biology, for synthetic biology, for predicting 3D structures. Then in the era of high accuracy, AI-driven protein engineering approaches, AlphaFold, Buzzy tool, which allows us to forecast the structure of protein with the accuracy very close to the experimental one, but this is not all the case when it works. But there is a number of other tools which can aid to what AlphaFold is not capable of creating on its own. Computation, so Deep ASNet and Rosetta and AlphaFold combination based on AI algorithms using computer vision approach to assess whether there are some changes to the structure of protein. Were they right? Does our computer model correspond to the reality? So this combination is used to particular changes to the structure of as protein for this as protein to become stable, in particular part with the biggest number of epitopes required to recognize real virus. We use several rounds. First round, there is a standard approach. You know, there is a region, hinge region. If you have two changes, such a confirmation is going to be more stable, and this approach is used. But instead of doing that, we forecast this segment of protein. The next stage, we decided not to focus on a hinge region. We used other regions of protein which can be replaced for as protein to become more stable in this conformation, in this state, and to improve immunogenic properties. In classification of proteins in the protein modeling, there are two approaches used. One of the approaches is about the structurized principle. When we move from the structure is a 3D sequence of atoms and molecular level with particular corners and mergers, and there is a different approach which is based on the following things. 3D protein structure is not that important. The first thing which is important is the sequence in protein, because they are coded in AIJ sequence, and this particular sequence is very similar to what we see in natural language. Moreover, if we take any amino acid residual, it's going to depend on the context it is in. Such algorithms, called sequence-based approaches, can be used to have additional optimization of S protein and to further improve immunogenic properties, to have more antibodies which will be efficient. To this end, in AI, there is an NLP process developed. In case of protein, we use the analog Wikipedia, UniPro, which uses 250 million protein sequences. It's UniPro database, and maybe mammal actually. This sequence is used to train a language model, and in scientific community there is a number of accessible models of effective transformers which can be used to be applied in transfer learning, and in this case to train language models to forecast homogenic properties of a protein. We use the approach which is about fine-tuning of pre-trained protein models. So we have looked into the number of examples how these proteins are recognized. We have collected models how the merges happen themselves. As of today, we have a lot of them, as protein is the mostly research system from the perspective of crystallographic data and understanding how antibodies recognize how they merge with S protein. We have a number of examples which is about mutation forecast, whether it's going to lower immunity and generated antibodies will be efficient or not. We trained 3 independent models, and we united in one model which we called immunogenicity estimation model, which allows us to understand if we have this mutation in a particular sequence, whether this mutation is going to be detrimental, whether the antibodies will be more efficient or less efficient in recognizing protein. To see whether this approach is efficient, we used a quite standard test, which is taking the data set for training and giving up all the S protein sequences, and to understand whether this model is going to represent what can be seen in experimental data based on the crystallographic structures database. Turned out that the model can find those segments of the protein which relate to epitopes. Red color relate to more immunogenic blue color to those epitopes where there is no recognition of antibodies, and we decided to check other systems where it are the 5 well-researched antigenes, and we checked whether the model can predict the sites of mergers and the number of efficient epitopes and the number of inefficient epitopes. That's not a unique model. There are a lot of examples, and in number of metrics we are at a winning position. In some of the metrics we're very close to those, but the main thing thing is that we managed to train the model in the way so it's in a quantity level can see how amino acid residue is going to be immunogenic and which is not. So in fact, the most important thing is the most important thing for the rational optimization of S protein. Therefore, we focused on the pipeline which we are developing for the virus S protein, but we are planning to do that and to expand that for other systems. So we're using two independent approaches. The first approach is based on this AI-driven structure-based optimization. Then the synthetic S protein optimized for vaccine, and then we put it in synthetic protein, and we expect this synthetic protein to have better immunogenic properties, which should be tested in real systems. Many approaches we are offering haven't been used before by other antigens. So some of the ideas might be more efficient, some of them might be less efficient. So soon we are expecting to get this data set and the protocol which would be more efficient, and it is possible thanks to a very close interaction with experiment and calculation models. So first of all, I'd like to thank our colleagues from Gamaleya Institute, which coordinate our computer and experimental activity, who work with experimental tests with a number of our algorithms, and our colleagues from Sber AI Lab and the Institute of AI. I'd like to apologize to colleagues which cannot be seen on the list. I'd like to thank the head of our group, Olga Kardeman. Thank you for your attention. Nikita, thank you very much. Very interesting, very important topic in view of the current situation. We have some time. Let me ask you one very important question. So, from your perspective, the stack of technology you have, the pipeline which you have created, is it scalable in terms of new threats? And how fast can we readjust to new challenges? How is it scalable? Is it difficult to adjust? Just in brief, your opinion, it's very important point. Our pipeline has a module structure. The modules are not based on a particular virus system, so it's quite universal and applicable to any virus, any threat which may emerge, and these approaches can be used in other systems as well, in case of other viruses, if they emerge. But the number of modules based on training, which was possible thanks to scientific community looking into pandemic, into the coronavirus infection, and then part of the pipeline cannot be applied at the same time, at the same moment. You need to accumulate the experimental data set for training. After that, you can train the pipeline models using a new data set and to apply for new systems. Nikita, thank you very much. I'm very glad that we have such developments in our institute, which is big motivation, not only among those people who are looking for new initiatives. I do really hope that your work will allow the scientific community to look into these problems. Through cooperation, we will create a faster pipeline which can be scalable in other instances. I'd like to thank Nikita once again.